So it's a pleasure to be here. I'm going to talk a little bit about some of the community-driven approaches that we're taking to really understand our genome, a little more focused on the clinical side of things. Make sure I know how to do that. Oh, there we go. So this is just a diagram of thinking about how the ClinGen project, which I'll talk about first, is orchestrated. It really starts with sharing data from patients, clinicians, laboratories, researchers, and then asking critical questions of that data. Is this gene associated with disease? Is this variant pathogenic? Is this information actionable for a patient? And through that process, really forming a curated genomic knowledge base that can be used to improve patient care. The first database that has been launched to support this project is Clinvar. Some of you probably know of Clinvar. I'll talk about it in a second. But there's other resources that are more gene-centric, things that we're building to also support this basic approach, an actionability sort of rubric for that information. A lot of the data can be found, or information is on our website. There's over 200 people involved in this project from over 75 institutions. So a lot of people working together on this effort. So I mentioned Clinvar. This is one of our key resources as part of the project. It is our variant repository, the place where we take submissions from the community and display them and provide them to the community. And that may be data from researchers, clinical labs, expert groups, directly from clinics or patients through patient registries, all of that data going into Clinvar. We also link to locus-specific databases where we can centralize data so we can provide it in easy download, but you can then link out to those databases for more detailed information. To date, there's been over 315 submitters to Clinvar, totaling over 172,000 variant submissions. So there's a lot of data coming in. Now, with all of that data from lots of different sources all making claims on variants, you might imagine there might be differences. And in fact, of over 118,000 unique variants with clinical assertions, about 11% of them have more than one submitter. Then we can compare those interpretations and really get a sense of how concordant we are in the community in interpreting genetic variation. And what you'll see is that we're not that concordant. You could look at this glass half full, glass half empty. Well, 83% of the time we agree, but 17% of the time we don't agree. And those are major disagreements between, pathogenic versus uncertain versus benign. So, and you can log into Clinvar, type in your favorite variant, and this is sort of the main screen per variant that you would see. And up in the top right is the overall clinical significance from all of the aggregated assertions from submitters. In this case, it says conflicting data from submitters. You can then go down and look at the clinical assertions tab and see who said what about that variant, when they said it. And that may help you to decide who to trust. Maybe one's really old and one's newer, and you could trust the newer one. But this is a way to at least have transparency in the community as to who's saying what. It's kind of like getting a second opinion from a second physician. So now, after we identify these differences, because we're aggregating all this data together, we can then start to resolve them. And this was from the first set of submissions that my lab and Emory in Chicago put in. Across our submissions in the first round, we had 104 differences. We immediately exchanged our evidence across all of those variants, and immediately were able to resolve 75% of those differences by just sharing the evidence that supported our conclusions. And we agreed with each other when we saw that data. That left about 25% of the variants that we still disagreed on with the same evidence. And then digging into those, it really came down to differences in our rules for classification, maybe frequency cutoffs for benign interpretations, et cetera, how we think about silent variants, things like that. When we worked through those differences and came to consensus on approaches, it left one variant we disagreed on. It was difference of opinion about a functional assay. We engaged experts in the community in that field, and we were able to resolve that one. So really, with a little bit of effort, came to complete resolution of all these variants. Now, it did take us some approaches on agreeing on how you classify variants, and recently we've come out with guidelines to now create more of a framework for consistency in interpreting evidence for variant pathogenicity, at least in Mendelian disease. This is the team that put this together. It looks like we're celebrating the completion of this guideline. In fact, that was the first working meeting when we realized the only way to come to consensus was with alcohol. And I'm not being, and that's actually true. There were a lot of things in this guideline that we ended up down to a vote because there really isn't necessarily truth out there that we can go with, and it did come down to expert opinion. And these guidelines only take us so far. ClinGen is now working on really building off of these guidelines, trying to create more quantitative approaches and evidence to support this. But we did at least have a starting point in terms of different types of evidence, how to weight them, how to combine them and come to categories. So we hope this will create more consistency in the community. And the other challenge is if we have thousands of variants coming in from all different sources, how do you know what to trust and what not to trust in a database like this? So we came up with this sort of tiered system where variants that come from practice guidelines, really well vetted, would get 4 stars. Expert panels and we approve applications for expert panel status. Some of the ones we've approved are shown there. They get 3 stars. And then and then dropping down to the single star, we now are implementing a new system to differentiate single submitters from those who provide their criteria, attest to a complete, thorough review of evidence, get 1 star, and their criteria are linked on the website. Will be in June when we launch this. The rest get no stars. If all the single submitters with correct criteria provided are consistent, it automatically gets 2 stars. So that allows this sort of aggregated set of data to define which one overrides the others when we're looking at aggregated assertions. And you can use the database to say, I only want the stuff at the top, and you can filter on that, et cetera. So it allows the community the ability to sort of pull out what you trust and what you don't trust and provide some of that guidance for it. Now, that's what all goes in, but if you look at the bottom, the question is, how do we work on resolving these differences and pushing things up that tier to the 3-star and 4-star? And we're working on, particularly some of the members of Carlos Bustamante's group here at Stanford are building our ClinGen KB knowledge base that is really a place that we can curate and move things up, resolve differences, et cetera. And so our clinical work groups, shown here at the bottom, can operate in that curation interface to try to move things up. But a critical thing is you don't have to wait for us to do that, to first at least get access to everybody's data that's being shared in Clinvar, and that's an important component of this project. These are just some of our clinical domain working groups that have been assembled. You can see Ewan on there in our cardiovascular group, the germline cancer group, inborn errors of metabolism, pharmacogenomics. We've just formed a somatic cancer group. I don't have pictures up there yet, but it's met a couple times so far. So really working through all that, expert curation. Now, one of the things that effectively informs a lot of the approaches we take is access to patient data, cases with diagnoses, sets of variants. That's what we all need to use to understand genomic variation. So how do we really build access to a case-level database so that we can inform our interpretation? And I think that will fundamentally underlie a continuous learning system that we need to create to support genomic medicine. So we've been working with a number of different models to support this. Most databases today are on the right side of this figure, or left on your side, like Clinvar, like dbGaP. These are centralized databases. People submit their data, and you get access through this centralized portal. Recently, we have a project undergoing the last couple of years called the Matchmaker Exchange. This is a truly federated network where the databases exist on their own, and then you connect through APIs to be able to share data. So it doesn't force everybody to have to send their data into a centralized place. The major use case for this is actually in gene discovery for Mendelian disease, where you may have 2 different patients with similar phenotypes, and you're trying to find the gene that causes these disease. You may have some candidate genes, so you found, like a de novo variant in a new gene in one patient, and that's not enough to implicate that gene in disease. You need that other patient around the world with the same phenotype, the same, a similar mutation, de novo mutation. How do you find that case? Well, there's lots of databases out there now. A lot of us came together 2 years ago or a year and a half ago at ASHG and said, let's figure out how to combine all our databases to be able to share these rare disease cases with candidate genes. And so we've also been working with the Global Alliance to put a lot of standards and consistency in the approaches that we were taking, and worked with each of the working groups in doing that. And recently, we've now launched the first iteration of that with 3 databases, one in the US, one in Canada, and one in the UK, that have launched the API, are now fully connected. So when you query with a candidate case from one database, you will automatically get access to the data in the other through a common API that's been implemented. So really exciting work, and we have an issue in human mutation that'll be coming out in September with an overview paper and about 12 other papers on all the matchmakers that are launching systems here. So fun stuff. Now the challenge with that model is that you have to then create links between every database. And there's a lot of work in doing that in terms of standing up all those linkages. Another approach is sort of the centralized hub model as a way to connect databases, but only have to create one connection to the hub, and then the hub manages all that interconnectivity. And we've launched such a system through our Gene Insight software, this share and share alike model called VariantWire, where all the groups that are using this particular software, their databases are connected in real time. To date, they're sharing over 32,000 interpreted variants. These are all clinical labs, so their interpretations are being shared in real time to support clinical assay and testing. We've also rolled out this same system in Canada to support all of the clinical labs in Canada being able to share all their clinical interpretations. And now we're bringing those Canadian labs onto the VariantWire network. Now, the only people who can access this data are people on this network, but we're working with each of the groups now that their data is better structured to help them submit it to Clinvar so that it's accessible to the public as well. So, and 4 of the groups have already submitted their data to Clinvar. So these are a lot of the approaches that we're taking. We're also in September launching a new structure that allows full genome database data sets to be connected to this sort of network. So when you query, you're not just getting clinically interpreted variants, but any variant across a genome in those cases with new search structures. So that's sort of the approach that we're taking to really take a lot of particularly clinical data and variant interpretation, try to support it through federated approaches to data sharing. And now I don't believe that there's going to be one database or one software that's going to support the world. I think it's going to be sort of a federated network of centralized hubs where each software system or EHR connects groups of people, but then they are federated across them. And in fact, this model is not new. This is how email works, right? Gmail connected to AOL, connected to Yahoo. It's a federated system of centralized hubs that allow us to send emails to each other when we're using different systems. So this fundamental approach, I think, can be applied in healthcare, will allow us to really share data in much richer ways. So I'm going to stop there, acknowledge a lot of people who are working on both the ClinGen project as well as the Matchmaker Exchange project that I mentioned, and this Gene Insight, and VariantWire network, and the COGR network in Canada. So thank you, and I think we'll take questions. Actually, we're going to move on to the panel, right?
Right. Thank you very much, Heidi.